Hey everybody, in this video we are going to talk about AAV in gene therapy, at least see how it has been used successfully to treat a genetic disorder. In the last video, I used this pencil right here, which is, a great pencil, and the tip doesn't break, but it's not as dark as these pencils here. So I'm sorry about that. I took a quick look at the video I put together, the last one, which was I think number 2501, and you can still see the writing. Even though my writing is kind of messy, but you can still make it out. But I'm back to these darker pencils for this video. So AAV in gene therapy. In the last video, we talked about just the general biology of AAV, adeno-associated virus. And essentially what we covered was it's an icosahedral virus, rather small virus, and it has a single-stranded DNA genome. I didn't mean to make the DNA like an S there, but a single-stranded DNA genome of about 4,700 nucleotides long. So that's going to put a limit on the types of genes that we can deliver to human cells with AAV. So it's probably going to have to be, relatively short. Well, shorter than 4,700 nucleotides. So, and what does the genome look like? Just a quick review. We have these inverted terminal repeats, ITRs, one at that end, one at the other end down here. I'll put a 3' end here, a 5' end here. This is a single-stranded molecule, and the structures right here are held together by hydrogen bonding, intramolecular hydrogen bonding. And there are two genes which together encode 4 different proteins. And we've seen in previous parts of the course how a gene can encode several different proteins based on alternate splicing, different transcriptional start sites, and things like that. So to use AAV in gene therapy, it's relatively easy, at least conceptually. We only need to remove everything between the ITRs and put the gene we want to deliver to, say, a human cell in place of the DNA we remove. So we're going to remove rep and cap, again, because we don't want to put a bunch of infectious AAV particles into a human. We want to put AAV particles that have the ITRs here and the transgene, whatever gene we want. As long as it is, we don't go above that limit right here for the whole thing, the ITR and the gene, this should work. Well, we should at least be able to make viral particles that can get into a human cell. And I'm going to say this is the transgene. And we know what genes are, but transgenes are essentially any gene that has been introduced into a genome using artificial means, and that's a good Wikipedia definition. So the gene we're going to be putting into human cells is going to be called the transgene. So scientists have figured out how to make trillions of these viral particles. So let's call these recombinant AAV particles containing the viral capsid. So the proteins that encase the genetic material, and the single-stranded DNA containing the ITRs and the transgene. So this will have everything that's needed to deliver that transgene into a cell, right? We talked about transduction, the transduction process with AAV, how it is taken into the cell by endocytosis, and then by mechanisms that aren't quite clear yet, the DNA molecule is delivered to the nucleus. So by initial studies of this process for use in gene therapy have found that this transgene with the ITRs tends to make its way to the nucleus of a human cell, and there, by mechanisms that aren't completely clear, becomes double-stranded DNA. So each of these is I'm showing you is the transgene. This is the single-stranded DNA molecule, including the transgene that has become double-stranded and formed into concatemers. So joined together, concatemers, as you take a single unit and you join them together, that makes concatemers. So, and those concatemers form circular DNA molecules. Circular DNA molecule, this is a circular DNA molecule containing many copies of the transgene and the ITRs. And scientists have found that these circular DNA molecules persist in nucleus of non-dividing cells. Non-dividing cells for many years. And what's a non-dividing cell? So there are many cells in the nervous system that don't divide. So in those cells, these circular DNA molecules containing the transgene will persist for many years. And how many? Why say many? Why not say one or two? Like how many years exactly? And we don't really know, because these studies are so new. It's hard to know exactly how long they'll last. So scientists we're left right now with thinking that, okay, well, maybe they'll last forever in a non-dividing cell. And what about dividing cells? Well, we don't really know that either, right? Because these might not be replicated. There's no replication origin here, but maybe we could incorporate that. Maybe that'd be something scientists can look at. And if you can get a replication origin in here, well, maybe these will be replicated and they can persist in dividing cells. But so far, they seem to persist for a long time in non-dividing cells. Okay, so let me summarize that. How to use AAV in gene therapy. So scientists have figured out all you need to do is replace the rep and cap genes with the transgene, get the transgene into the viral capsid here. So we won't get over the details of how that works, but scientists can make trillions of these things, and we can get those, and then we can inject intravenously into a patient to deliver that transgene into the patient's cells. And in the patient's cells, the transgenes are thought to form concatemers and circular DNA molecules that persist for many years in non-dividing cells. So, and I should say that I think all this data right here is known from studies of animals. So this is what we think is going to happen if this process is used to treat a human genetic disorder. And it has been used in the treatment of spinal muscular atrophy. So this is a pretty terrible genetic disorder. Essentially, it results, there's a number of different forms. We're going to talk about a number of different forms in the next video because we have another treatment that can also be used for this genetic disorder that does not involve gene therapy. It's something else, and it came out right about the same time. But for this one, just knowing about SMA1 is good enough for understanding how this gene therapy works. So SMA1 results from mutations in the SMA1 gene. So most often results when parents are carriers. So each parent is heterozygous for a functional copy of SMN1 and a null allele of SMN1. And there's a 1/4 chance of them producing a child that is homozygous null for the SMN1 gene. And this genotype has a survival likelihood of essentially less than 2 years. So the muscles become very weak, and survival is usually less than 2 years, or at least mechanical ventilation is necessary before the 2 years of age. So, and then life doesn't extend after mechanical ventilation is necessary. The prognosis is very poor once mechanical ventilation becomes necessary. So it's a terrible disease. And okay, so in 2017, scientists and physicians published a study where 15 SMA1 infants of about 5 months of age, some were a few months younger, some were a month or two older, were injected intravenously with AAV serotype 9, recombinant AAV9. So the rep and cap genes have been removed, so it's no longer infectious. And the functional SMN1 has been put in place of the rep and the cap genes. So they delivered 50 to 200 trillion viral particles. So 50 to 200 trillion of these, of the AAV9 carrying the SMN1 gene, per kilogram of body weight. And about six months old infants are on average 5 to 10 kilograms. Whoa, so you can see that's a lot of virus. There's a lot of recombinant viruses that they put into, that they injected intravenously into these infants. So in the notes, you should check out the notes here. I put a couple tables from this study. What is the title of the study? The study is Single Dose Gene Replacement Therapy for Spinal Muscular Atrophy. And I think it was the New England Journal of Medicine in 2017 where this was published. And you know, there are copyright issues. I don't know if I can actually show the figures in a YouTube video. So I'll just describe it, and you can take a look in the notes so you can actually see the table for yourself. Actually, you can look at the paper too. But in figure 1, they have a figure where they show results for each of the 15 patients. They have 2 cohorts. Cohort 1 was giving a lower virus titer, so I think like 50 trillion, I should say, particles per kilogram. In cohort 2, I believe, was closer to 200 trillion particles per kilogram. And down here on the x-axis, what they showed was the data from historical cohorts. Now, this historical cohort, what that is you look through medical records and you see what is the normal phenotype or disease progression in patients with spinal muscular atrophy type 1. I'll change that to SMA1. So these are the different ages: 10.5, 8 months here, 10.5 months here, 13.5 months here, 20 months here. And down here, what they're showing you at 75% of individuals in the historical cohort who had SMA1 did not require mechanical ventilation at 8 months. 50% did not require it. It means 50, that also means 50% required it by 10.5 months. So we have 25% here at 13.5 months and 8%. So only 8% of infants with SMA1 were free or could get by without mechanical ventilation at 20 months. But every patient in this study was breathing on their own past 20 months. So, oh, that seems really amazing, right? And do I have to draw all these lines? I don't think so. I don't think so. But I can't stop. So I'm going to finish. Okay, so, wow, okay, 100%, we're doing, relatively great, comparatively great compared to individuals who did not receive the gene therapy treatment. So figure 2, what was going on in figure 2? So this is neurodegenerative disease or disorder, and motor functions really don't progress in infants and toddlers with this disease. And in figure 2, what they do is they do a test which kind of quantifies motor ability in developing children. And let's say this is the 5-month time point right here, and this is 5 months right here. And this data over here, this is going to be cohort 1. Over here, this is cohort 2. And what they show is that for the 3 individuals in cohort 1, well, none of them reached this. This is a score of 60 up here. This is some test called, I think, the CHOP test, and CHOP stands for something, Children's Hospital of Philadelphia, I think, or something like that. And I guess children with SMA1 almost never go above 40. And the patients in cohort 1 didn't quite get above 40, but every patient in cohort, I think every patient but one in cohort 2 seemed to be doing quite well. And the difference between cohort 1 and cohort 2 was mostly the viral titer, so the number of viral particles injected intravenously into the patients. And while the 3 patients in cohort 1, I guess, were treated after 5 months, most of the patients in cohort 2 were treated early, before 5 months. So this data suggests that gene therapy should be started as early as possible, with a viral titer closer to 200 trillion viral particles per kilogram of body weight. So far, gene therapy seems to be working really well for the treatment of SMA1. Now, that was a study that was published in 2017, Science Magazine. You can see this in the notes also, some excerpts from the magazine, and also a link to the article so you can read it on your own. Science Magazine did a write-up on this trial, and they describe an individual, a young girl whose parents were carriers for the disease, and she had an older sibling who was born with the disease and passed away. But the parents decided to have another try at a child, and 1 in 4 chance of EV, I'll say, inheriting the disorder, right? 1/4 chance of being homozygous null for the SMA1 gene, and really unfortunate. Yeah, so she inherited SMA1 too. So she was entered into the trial, and they show pictures of her, 3 years old, and they describe her as being really no different than any other 3-year-old. So the data I kind of went over was like, oh yeah, okay, so they're doing better than most patients that don't receive a treatment. But the article in Science Magazine suggests that, oh, this is a miracle treatment, and there's no difference between EV, the child here, and I lost my train of thought. I must have low blood sugar right now. Okay, what was I saying? So, okay. Anyways, I encourage you to read this article. I've been nervous to see how this little girl is doing. It's been about a year since I looked up or tried to do a follow-up and see if the treatment is still working and if she still is leading a healthy life, free of the effects of this disorder. So I have been nervous to look up information on that, but so far so good. So far so good. It looks like gene therapy is working in the case of SMA1 for these 15 patients. Okay, so in the next video, what we're going to do is take a little bit closer look at different SMA, spinal muscular atrophy subtypes, and a new treatment that's not gene therapy. It's based on ASOs, antisense oligonucleotides, and I think it's called nusinersen is one of the names of the pharmaceutical that is used to treat individuals with SMA1. And when this gene therapy treatment came out, physicians were at a loss because they both looked good, but they didn't know which one would be best for their patients. So they were having trouble deciding, well, is gene therapy going to last forever, or should we go with nusinersen? And you know, I'm not sure if they figured out which one is better. I know one of these treatments costs like $1 million. I think they're both very, very expensive. But anyways, we'll talk more about that in the next video.